Hi, everyone, and welcome to today's lecture on translation and proteins.
Now, to remind you with translation, again, you're going from nucleic acid to protein. And in this process, mRNA is the code that will basically tell the ribosome what amino acid needs to be where. rRNA is very important. That's actually part of the ribosome. And the fact that the ribosome has some rRNA, which is nice, clingy single strands of RNA, that allows the ribosome to grab a hold of the mRNA to then use it as the template. You then have tRNA, which is going to be what's actually holding the amino acids and brings them together so that the ribosome can build the protein. Now with tRNA, sometimes you'll hear it referred to as charged or uncharged. Charged simply means that the tRNA currently has an amino acid, whereas uncharged means that the tRNA gave away its amino acid, so it's no longer holding any amino acid. Now, one other thing I want to remind you of on this slide is the fact that this ribosome structure over here, which, like I said, that has some rRNA on it. So this ribosome structure is what we call a nucleoprotein. Nucleoprotein is important because, again, the fact that the ribosome is protein because it's an enzyme, but also has some nucleic acid, the rRNA, allows it to grab to grab mRNA, which is important. Now, the ribosome has 3 sites on it. It has A, P, and E site, so you could remember it as APE or EPA. The A site is the amino acid acceptor site. So the tRNA that's in an A site will have just a single amino acid. It'll be charged. The P site is the peptidyl or peptide site. So the tRNA that's sitting in the P site will have the growing polypeptide string, the growing protein. And the E site is the exit site. So that's where the uncharged tRNA will then leave the ribosome. This will become much clearer in a minute when you review the video. Okay, this slide is just so that you can pause the video and write down the notes in case you didn't catch what each of the sites were.
So speaking of that tRNA, I want to first of all remind you that if we say that tRNA is charged, that simply means that the tRNA is currently holding an amino acid on it. Okay, so it's charged if it's holding an amino acid. If there's no amino acid, we call it uncharged. Okay, now when the tRNA is getting charged, when you're charging it, it means you're adding an amino acid to it, and it ends up occurring in a two-step process, which you see over here. The first step involves the amino acid reacting with ATP to form what's called the amino acid AMP. Then in the second step, you have the amino acid group transferred to the tRNA, okay? And so this crazy little structure here that you see, that's the tRNA. Now, in addition to the tRNA being critical and the mRNA being critical, the ribosome is also critical. And I want you to circle star highlight that bold red term, nucleoprotein. So nucleoprotein is what it sounds like. It's a nucleic acid linked to a protein. In the case of the ribosome, the nucleic acid is a small piece of RNA. And we're going to talk about that more as we go through the video and the actual process of translation. Now, nucleoproteins are very, very significant because the fact that it has this little piece of nucleic acid, in this case, a little piece of single-stranded RNA, well, what do you know about single-stranded nucleic acids? If it's single, it's clingy. It wants to find its match, don't we all? And so now what happens is because the ribosome has this little piece of clingy single RNA, that allows the ribosome to stick to or to grab other pieces of matching RNA. Okay, and we're going to see that when we look at the process of translation. So this little piece of RNA on the ribosome will allow for that ribosome to grab the mRNA. And that's a critical part of initiation and of the whole process of translation. Now, the ultimate role of this structure is to synthesize proteins, to stick all of those amino acids together. Now, you'll notice that when you look at ribosomes, you can either see 50S, 30S subunits, and that's for prokaryotes, or eukaryotes are somewhat larger. They have 60S, 40S, but I'm not going to make you memorize any of these. And it's always funny because students think that they made a mistake in the figures and all because it's not additive. So you see 50S linked to 30S and suddenly it's 70S instead of 80. Don't worry about that. It's not an additive type of procedure, and I'm not going to make you memorize those numbers. So now we're going to get into the process of translation. And just like when we talked about transcription, there's going to be three main steps. There's going to be initiation, elongation, and termination. Okay, so now first we're going to think about initiation and elongation. So what you'll notice is a few things. The first thing that we're going to need is you're going to need what's called the Shine-Dalgarno sequence. Okay, so the Shine-Dalgarno sequence will be on the mRNA. It'll be near the 5' end, but this is the Shine-Dalgarno is part of every mRNA that needs to be translated into protein. So each mRNA will have a little Shine-Dalgarno sequence. This complements that little piece of RNA that I said the ribosome has because the ribosome is a nucleoprotein. Okay, so the Shine-Dalgarno will be on the mRNA and the anti-Shine-Dalgarno is part of the ribosome. So this allows the ribosome to now grab the mRNA. And when you look at the mRNA, so we have the Shine-Dalgarno and the anti-Shine-Dalgarno, okay? When you look at the mRNA, you'll see that you eventually get to the start site of that mRNA, AUG. That matches the anticodon of the tRNA that holds FMET. Okay, so each protein, the first amino acid, will be formal methionine if it's a prokaryote or just methionine if it's a eukaryote. And that'll be the first one. So because the initiator tRNA is charged with methionine or MET. Okay, now when you look at the ribosome, normally tRNA holding an amino acid will come in at the A, the acceptor site. Okay, and then the P site usually has the tRNA that has the growing polypeptide. But in initiation, the very first tRNA, the initiator tRNA, which is the one holding or charged with methionine, that's the only tRNA that's going to come in first to the P site. Okay, so the methionine initiator tRNA comes into the ribosome at the P site. The second amino acid charged tRNA will come in and sit on the A site. When they're now sitting next to each other, I want you to make the pa sound, okay? Or since, a lot of you are considering the medical field and always think of PAs, a lot of you want to be a PA, think of PA. The reason I say this is because the transfer that occurs to make the growing polypeptide, what happens is the amino acids that are currently sitting on the P site get cut off of their tRNA and they get linked to the amino acid that's at the A site. So transfer is from P to A, the growing polypeptide to the one sitting on A. In this first initiation step, it's methionine is currently at the P site. The second amino acid comes in at the A site. So the methionine is cut off of the initiator tRNA and it's transferred to the tRNA and the amino acid at the A site. Okay, P to A. Circle, star, highlight that in your notes, please. Okay, what then happens is the ribosome shifts. It slides over so that each tRNA is now where it's supposed to be, meaning that the empty uncharged tRNA is now at the E, the exit site. The one with the growing polypeptide that was just in the A site is now shifted over to the P site, the polypeptide site. The A site is now open so that the next amino acid can come in, okay? And that keeps going in elongation. Okay, you're going to see it in a moment in the video, but I just wanted you to start to get comfortable with that process. But again, make sure to keep going pa, that PA sound.
To extend a growing polypeptide chain, the ribosome must select the correct amino acids that are specified by the messenger RNA. An aminoacyl tRNA bound to elongation factor TU, EFTU for short, enters the free A site on the ribosome. If the anticodon of the charged tRNA does not match the codon in the messenger RNA, the tRNA is rejected. The process of trial and error repeats until the correct tRNA is identified. Elongation factor TU hydrolyzes its bound GTP and dissociates. If the tRNA is correctly matched and remains bound for a long enough time, it is committed to be used in protein synthesis. The ribosome catalyzes the formation of the new peptide bond and undergoes a dramatic conformational change. Elongation factor G binds to the ribosome. Hydrolysis of GTP by elongation factor G switches the ribosome back to the state in which it can accept the next incoming tRNA.
In life, everything must have a beginning, a middle, and an end. And so when we talk about translation, the end is termination. And the reason you end up with termination in translation is because there are three possible stop codons. Now, what makes these termination is that there are no tRNAs that can read these codons. And what that means is there is no tRNA that has the anticodon that matches these, which means that there's no tRNA that's going to be able to come along and stick to this spot to bring in any other amino acid. Instead, what reads these stop codons are called release factor proteins. So the release factor protein binds to that spot instead, and it basically will activate. So once it's bound to that spot, it will activate peptidyltransferase that's going to cut the bond between the polypeptide, so the protein, and the current tRNA, the very last tRNA that's on the ribosome at that moment. Okay. At that point, what then happens is the ribosome is taken apart, and that is so that it could then be recycled. So it's conserving energy and resources to then recycle the ribosome. Now, I want to point out when we talk about translation and mRNA, there's actually some variations amongst mRNA. So, eukaryotic mRNA, they each code for a single protein, whereas prokaryotic mRNA often carries several coding sequences, meaning that you'll have a bunch of coding sequences on that one RNA, and then you end up getting multiple proteins out of it. And so that's something that we talked about in other lectures as the idea of monocistronic eukaryotic uh, systems versus prokaryotic polycistronic. Okay, so you can think of that PP. And I'm sorry, no matter how old I get, I still laugh at that. But yeah, that'll help you remember polycistronic for prokaryotes. And so there's several coding regions. Now, what's really interesting and a unique difference between prokaryotes and eukaryotes when we talk about proteins and translation and all, I want you to circle star highlight the fact that prokaryotes can do what's called coupled transcription translation. And that's what you see in this figure here. So basically, you have DNA coding for the RNA, and before that RNA even finishes getting synthesized, you have ribosomes jumping on and starting to read it. And as they read it, they're starting to produce proteins before the RNA is even fully finished yet. And that's unique to prokaryotes because think about it. In order to have this happen, you need to have DNA, RNA, and ribosomes all in the same place. You can have that in prokaryotes because what don't they have? There's no nucleus. So prokaryotes do not have a nucleus that separates the ribosomes from that DNA while it's making RNA. Okay? Eukaryotes, on the other hand, our cells have a nucleus. So the DNA and the RNA are held together in that nucleus for transcription, whereas translation then has to occur outside of the nucleus where the ribosomes are. So in eukaryotes, you need the DNA to fully finish coding for the RNA during transcription. Once that's finished, the RNA then gets transported out of the nucleus and heads toward the ribosomes. Okay, very important that can the difference between prokaryotes and eukaryotes when it comes to that process. So please make sure you circle star highlight this question and understand the concept. You can message me in the Remind app if you need a little further clarification, but it's very, very important difference. So, in addition to the differences that we just talked about with mRNA and with coupled transcription translation, I want to point out that the overall process of protein synthesis is very similar between eukaryotes and prokaryotes, but there are some differences that pop up along the way. The big one that I want you to circle star, highlight, and make note of is that for initiation, eukaryotic mRNA is recognized by the 5' cap of the mRNA. Eukaryotes do not have a Shine-Delgarno sequence. So I want you to put stars next to that. Initiation in eukaryotes, the ribosome will grab the 5' cap of the mRNA, whereas in prokaryotes, the ribosome will grab the Shine-Delgarno sequence of the mRNA. I also listed out some other differences, and you have this big table here showing the overall differences, but I don't want you to worry about memorizing or trying to figure out all of these differences. This was just to show you that ultimately eukaryotes, they're basically more complex. And so all of these differences that you will see kind of accommodate for that. But I want you to focus on the difference of the 5' cap versus the Shine-Delgarno. Now, when we think about protein synthesis, if you think about the cell, 2/3 of the organic matter in a cell are proteins. And so synthesizing the proteins can end up consuming a lot of time and energy and resources or materials within that cell. So you need a way to be able to halt protein synthesis and conserve resources in the cell. Now, prokaryotes and eukaryotes do this in two slightly different ways, based on what they're targeting. Prokaryotes, what they'll do is they will only transcribe genes that are essential to conserve, and they will, save even more time, energy, resources by not even making the mRNA a lot of times. They will also, when it comes to targeting something, they will inactivate ribosomes. And so what they do is they turn the ribosome into a dimer. And so when the ribosome is a dimer, picture it kind of stuck together, um, you can't have protein synthesis. And then when conditions are favorable and the cell wants to synthesize the proteins, it'll then disassemble that dimer. Eukaryotes, on the other hand, they target the elongation factors, which are basically, they're targeting elongation or even initiation factors that would end up using energy to function. And so they phosphorylate them and prevent them from being able to work. And if those can't work, if the elongation factors can't work, then you can't have protein synthesis either. Okay? So prokaryotes target the ribosome itself, whereas eukaryotes, you can circle the E of eukaryotes to remind you that they target the elongation factors. Now, once the proteins have been made, they're not going to be used exactly where they're made, which means you need some sort of way to have the protein exported or transported out of the cell. The way cells can do this is there will be a signal sequence at the N terminus, and that sequence gets recognized by what's called a signal recognition protein. And that will guide the target to what's called the translocase complex of the cell membrane. So over here, you see the signal recognition protein will come along and it will guide the newly made protein to the translocase in the cell membrane, okay, to transport out the protein. And then once that protein is outside of the cell membrane, that signal gets cut off by a peptidase. Okay. And in eukaryotes, the transport can either be across the endoplasmic reticulum membrane or across the plasma membrane itself. Okay. And in eukaryotes, you have the Golgi for packaging and secretion. Okay. The Golgi apparatus is for packaging and secretion. In addition to transport being important after a protein's been synthesized, what is absolutely most important is then folding that protein. Because as you know, structure defines function. How a protein is folded is critical to it actually working. So now, in order to oversee that occurring, we have what's called chaperones, okay, chaperone proteins or chaperonins. There are two categories. One category will prevent premature folding as the protein is getting synthesized. The other one will correct misfolding. So here you see the two mechanisms. In this first one, you have the ribosomes synthesizing the protein. The yellow structure is the protein, yellowish green. And that HSP70 is a chaperone that will bind to the newly synthesized protein. And it almost looks like when we talked about single stranded binding protein and DNA replication. By having this all bound to that new protein, the protein can't fold yet, right? Because right now the amino acid residues, they're not exposed to fold because the chaperone's blocking that. Okay. Once the peptide passes through the translocase and is where it's supposed to be, then the chaperones can pop off. And now naturally that protein's going to want to fold because we talked about all those R groups that like to interact with each other. The second category over here, this is for when a protein has unfolded, okay? It's denatured, it's unfolded, which can be a real problem because, you have that unfolded protein floating around. Other proteins are going to want to interact with it because now all those beautiful, luscious looking R groups of the amino acids are exposed, right? And so they're going to want to start interacting with that protein. So chaperones that are, such as the Grow E type, they will basically provide a sequestered environment for that protein to naturally fold on its own without any other proteins coming along and trying to interact with it. So it gives it a chance to refold properly. Whereas chaperones or chaperonins are responsible for folding proteins, I want you to remember that protease is responsible for degrading proteins. And when you think about it, we said 2/3 of the cell's organic matter are proteins. So you can't have every protein that has ever been made stay in the cell. You need a way to degrade proteins. I tell students to picture it in the funny, dramatic way of if you had every protein stay forever in the cell would just explode. It would be massive. So you have to have a way to degrade the protein. So we have proteases that are enzymes that degrade proteins. But now the problem is, if these are enzymes that degrade proteins, and I just said 2/3 of the cell are proteins, and we know that they're important proteins in there, proteases are dangerous. The problem is, if they degrade proteins that you need, and it's not the time that you want those proteins degraded, you'll end up killing the cell. So the solution to that is that proteases need to be highly controlled. They can either be held in separate compartments, like a proteosome that just houses those and will take care of degrading the proteins, or they can be designed so that they only recognize tagged proteins. Proteins, for instance, tagged with ubiquitin. Okay. The other way that they can be controlled is that they can sometimes be synthesized as inactive precursors, and then they get activated once they're outside of the cell so that the cell is protected from their activity. To give a little more detail on degradation of proteins and kind of give you some notes on some of the terms that I mentioned in the previous slide, eukaryotes for degradation of proteins have what's called proteosomes, and the proteosome is a cylinder that's chock full of proteases, and so it has protease active sites. And basically, what happens with the cylinder is the top and the bottom will recognize and bind any of the damaged or the misfolded, denatured proteins, and it will bind them and kind of pull them in. And proteins that are degraded, they're going to be tagged with what's called ubiquitin. It's a short little amino acid sequence, sequence of amino acids. And so when you have the ubiquitin, those are on the protein, those are partially what's helping those, top and bottoms to recognize and bind the proteins that need to be degraded. And so they get pulled in, they get chopped up, the proteins, and then the ubiquitin, which here is highlighted in a different color, the ubiquitin ends up getting cut off and recycled so that that ubiquitin can then go tag other misfolded proteins, and the cycle continues. So you have ubiquitin tagging a protein that has been denatured or is not functioning properly. Once the ubiquitin tag is on that misfolded, denatured protein, it could get recognized by the proteasome, gets pulled in, and the proteases in there will chop up the protein and also cleave off the ubiquitin to get recycled back again. Okay? Now, as I keep emphasizing, structure and function of proteins is critical because structure determines function, and the function is everything. Okay, that's what's critical about proteins. Now, I keep mentioning how important structure and folding is for proteins. So I just want to point out that if proteins are not folded properly, you could end up with very serious diseases, and these can include things like cystic fibrosis, Alzheimer's, mad cow disease. And when we talk about these dysfunctions or these diseases, I want to point out that there are two terms that you hear when it comes to protein structure, and it could either be proteolysis or denaturing, two very different things. So I want you to know that for proteolysis, that will destroy all four of the levels of structure that we just talked about, including the primary structure, because in proteolysis, you are chopping up the protein. So even the initial sequence of it, you've now ruined because you've cut it into little tiny pieces and they're all floating around separately. Denaturing, on the other hand, is simply unfolding the protein. So you do lose secondary, tertiary, and quaternary structure. But what don't you lose is the primary structure because the whole protein is still intact. The sequence of amino acids are still in the proper sequence, they're just not folded anymore. And so that can be reversed, that's reversible, whereas proteolysis is irreversible. Now, we mentioned how important protein structure is in the folding of proteins, but what's also very important with protein function is that they have extra little non amino acid components, meaning things that are not proteins are attached or floating around with the proteins to help them activate and to help them function. One of the terms I want you to know for that is the term cofactors. And I always have students think factory when you hear that. Factory meaning, picture some steel factory. It's inorganic. Cofactors are inorganic molecules that will help to activate enzymes and that will help proteins function. Now, they are free floating, whereas if, let's say, you have a metal ion that's fixed, that is permanently attached to the protein, we call that a prosthetic group instead of a cofactor. Now, with these associations of cofactors, prosthetic groups, also you'll hear a term coenzymes, which is when the molecule is organic, such as vitamins to help activate an enzyme or a protein. You'll hear the terms apoprotein and holoprotein to kind of give you information about the protein, whether things are associated or not. When you hear apoprotein, that is just the protein. It does not have any of the cofactors, coenzymes, no prosthetic groups, nothing. It's just the protein subunits itself, which means that it's inactive. Okay, so an enzyme that's an apoprotein form is inactive. Whereas holoprotein, I always do the highly offensive accent of my people, the Italians, and I say, think of holoprotein. So when you see holoprotein, think holoprotein is the whole thing. It has the cofactors, it has the prosthetic groups, metals associated with it, plus the protein itself. It is active. It's the whole thing put together. The example that I like to have students remember is heme of hemoglobin, where you have the protein, but you also have the central iron that is going to bind the oxygen. And so that's what you see in this picture here. So this here is the holoprotein. You have the protein part and you have the metal ion prosthetic group. And I apologize again for how horrible that accent is of my ancestors. Now, in addition to the associations we just mentioned, a lot of proteins are actually conjugated proteins. And what that means is that a lot of times there are special proteins that are linked to other molecules. And if you look at the name of the protein, that helps you figure out what molecule it's linked to. So the first one is glycoproteins. Glyco or glycans is another word for carbohydrate. So, a glycoprotein is when you have a protein and a carbohydrate linked together. They're frequently found on cell surfaces. So, for instance, you see here protein with sugar residues on top. And that can be very important for things like cell signaling and antigen recognition and a whole bunch of other stuff that we'll talk about in other lectures. You also can have lipoproteins, and lipo meaning lipids means you have a protein plus a lipid. A big example of that is beta lactamase. And beta lactamase, for those of you who like microbiology or who have ever had to take antibiotics, beta lactamase can be very, very valuable to protein, to bacteria, because this protein, this enzyme, allows the bacteria to cleave a beta lactam ring. Now, where do you find a beta lactam ring? In penicillin and penicillin family antibiotics. So this enzyme here actually gives bacteria antibiotic resistance. So if you've ever had to take antibiotics and they didn't work and you had to take a different cycle of it, one of the things you might have to blame is beta lactamase, a lipoprotein. The last one over here is a nucleoprotein, which you guys are already experts at. That is when you have a protein plus nucleic acid, and the two big examples I want you to remember are the ribosome and the spliceosome. And both of those are nucleoproteins in which it's protein plus RNA. Okay, so all of these are very important and valuable to us. Glycoproteins, you see that in cell to cell adhesion, cellular recognition. For instance, sperm recognizing the egg, even in your immune system reactions. That's glycoproteins are critical. Lipoproteins, we said, involve things like antibiotic resistance. And nucleoproteins include ribosomes, spliceosomes, all the things we need to make proteins to make mature RNA. So conjugated proteins are very, very important in the cell. The last thing I want to mention in today's lecture is the concept of post-translational modifications of proteins. So this means that after a protein has been made or synthesized, modifications can be made on some of the residues. Now that's important because if you think about it, we always say, structure defines function. So if you modify the structure, you've now modified the function of that protein. And it could be very important for tag purposes or for folding purposes or cell signaling, various different reasons. Now, some of the common modifications include things like phosphorylation that you see here, methylation, which is over here, acetylation, ubiquitination, even things like glycosylation and lipid modification, sort of like the conjugated proteins that we just looked at a moment ago. And when these modifications occur, a lot of times it tends to be particular amino acids that are most commonly targeted by particular modifications. So, for instance, when we talk about phosphorylation, the main residues that can be targeted are histidine, aspartic acid, arginine, serine, threonine, and tyrosine. And I'm going to have these listed out on the chalkboard note slide. I want you to know um, for the modifications that I mentioned, which amino acid residues tend to be what's modified. The next one I want to mention is acetylation. I want you to circle star highlight that lysine is the main residue targeted. And a lot of times you can also see protein amino termini targeted. But lysine acetylation comes up a lot in research and medical implications and whatnot. So I want you to really remember that one. Also, another interesting one is glycosylation, which we don't have in this figure here, but glycosylation tends to be with asparagine targeted or arginine, serine, or threonine. And then some of the other modifications that occur, it tends to be one particular amino acid that gets targeted. So in lipidation, it's cysteine. In acylation, it's lysine, which we just saw with acetylation. In thiolation, so thiols, we mentioned in previous lectures how significant thiol groups are. That is cysteine, of course. And, think about cysteine, always think disulfide bonds, protein rigidity, very significant amino acid. And then oxidation, I want you to know methionine. And that is it for today. As always, if you have any questions or concerns, please send me a message in the Remind app. And always make sure to send me a picture of the exact slide that you need help with, because that will help me to better answer your question. Thank you and have a great day.